Okay, so I didn't put my email address there, but on the last slide, my email address. So if you do have any questions, you can either shout out during the lecture or you can email me afterwards. So as you can tell, we're going to talk about B cells and antibodies. And on Thursday, we're going to look at the other side of the adaptive immune system, which is the T cells and the MHC. So the adaptive immune system is very different to the innate immune system that you've already had lectures on. Okay, you've had a couple of lectures on the innate immune system. Today we're going to look at the first part of the adaptive immune system. And the adaptive immune system is essentially what the lymphocytes do. Okay, there are two kinds of lymphocytes. There's B lymphocytes or B cells and T lymphocytes or T cells. And they talk to each other. There's a little diagram here. We're not going to go into any detail now, but what we're going to spend the next couple of lectures looking at is that interaction between T cells and B cells. Okay, and we're going to start off today with the B cell side of things. It's quite difficult to separate the two in actual fact, so we'll probably be touching on T cells a little bit today, but we'll go into it in more detail on Thursday. So like I said, the T cells and the B cells are a hallmark of the adaptive immune system, and they're very different the way they respond to the innate system. So the innate system you've heard about, the recognition of foreign bodies, pathogens by the innate system is mediated by pattern recognition receptors. You remember those? Things like toll-like receptors. So those are receptors that are sort of hardwired into the genome. There's very little scope for them to change. And it's quite easy, actually, for a pathogen to sort of evolve away from recognition by the innate system. So the innate system really is only the first line of defense against a pathogen. It's the adaptive system that comes in later that really affects the removal of the pathogen. So the adaptive system uses antigen receptors. So both T cells and B cells have antigen receptors. And the diagram here of those antigen receptors. Here's a B cell antigen receptor, and here is a T cell antigen receptor. And you'll see by the color coding here that in actual fact, the T cell receptor looks like just one of these arms of the B cell antigen receptor. So we're going to look at the B cell antigen receptor today, and just for future reference, we're going to talk about these pieces. This part that's colored here is called the FAB fragment, or antigen binding fragment, and down here we've got the FC fragment domain, which is called FC because this crystallizes quite readily. So the key difference between antigen receptors and pattern recognition receptors is that there are billions of different antigen receptors, billions and billions, and that together, that a collection of different receptors is called the repertoire. And it's different to the innate system because these antigen receptors are not hardwired into the genome. The gene segments are in the genome, but they only come together in the maturing lymphocyte. So these gene segments are produced, so they're rearranged to produce a functional antigen receptor as the lymphocyte develops. Okay, so that's very different to an innate receptor. So, those are expressed in dendritic cells and macrophages and all the rest of it. An antigen receptor is actually generated by a process of gene rearrangement as the lymphocyte develops. So this process is completely random, as you'll see, and it generates a lot of different antigen specificities. Each receptor has a unique antigen specificity. That's how it is able to recognize a plethora of different pathogens out there in the environment. But it has no capacity to distinguish foreign from self. Okay? Pattern recognition receptors, they recognize things that are just peculiar to pathogens. Things like, unmethylated single-stranded DNA or double-stranded RNA, those kinds of things. Those are, recognized by toll-like receptors. But the antigen receptors don't know the difference between self and non-self. So there has to be this process where all of those autoreactive lymphocytes are actually removed from the repertoire. So the way that the adaptive system discriminates self from non-self is by removal of all of the autoreactive lymphocytes. And what are left essentially lymphocytes that have receptors for foreign bodies. Okay. The other thing that we need to talk about is that because these antigen receptors are essentially unique, each lymphocyte has its own sort of unique antigen specificity. Each lymphocyte is quite rare in the repertoire. And so whenever it is required for an immune response, that particular lymphocyte has to proliferate. We call it clonal expansion. Okay, so these receptors are clonally distributed. In other words, each antigen specificity is on a single lymphocyte. And in order for that lymphocyte to be able to effect removal of a pathogen, it has to expand that clone. It's called clonal selection by antigen. The antigen picks the clones that respond to it. That lymphocyte then proliferates, and that's called clonal expansion. That process takes time. So if we look at a typical immune response shown in this little diagram here, so what we've got here is the production of an antibody up the side here. And on the bottom, we've got time. So if we're exposed to a particular antigen on a pathogen, or a vaccine, for example, you don't see very much for the first few days. This is because those lymphocytes are gearing up, they're proliferating and differentiating. And that takes time. And you don't start to see antibodies. This on the left here is antibody in the serum. You don't start to see antibodies for a few days. Just around a week, you start to see the appearance of antibodies. And it's during this lag phase that it's called, that the innate system is kind of holding back the infection. Okay, so the innate system is your first line of defense, and it provides that protection while the adaptive system is getting mobilized. And then, of course, you start to see the appearance of antibody in the blood. It starts to increase in titer. This is a log scale. And then it will reach a plateau. Those antibodies and the T cells will start to affect removal of the pathogen. Gradually, those lymphocytes are killed off because they're not needed anymore. And gradually, the titer of the antibody goes down again. And you'll have sort of a baseline of antibody that's a little higher than it was before, but it's maintained at a slightly higher level. And one of the interesting things about the the adaptive system is that some of those lymphocytes are left behind after the response, and they're called memory cells. So we have memory B cells and memory T cells. We also have long-lived plasma cells. The long-lived plasma cells are actually the B cells that make antibodies. We'll come to this in a minute. But some of those plasma cells remain after the immune response has died down, and those are responsible for sort of maintaining a slightly higher level of antibody than there was before. So the second time you get exposed to the antigen, you get a much quicker response. Those memory cells, they can be activated very quickly, and the antibodies are produced much more rapidly. In fact, so rapidly you don't even see any disease symptoms. When you have a primary response, there's a window of opportunity here for the bug to cause disease symptoms. But once you're exposed a second time, those memory cells and the antibodies that are left over from the previous exposure can knock down the infection before you even see an infection. You'll notice that the response to an unrelated antigen is just like a primary response. So because these receptors are very antigen specific, those memory B cells and memory T cells are very antigen specific. So that memory only really applies to the antigen that it saw in the primary response. If you get exposed to a different strain of flu, for example, then you have to mount a new response all over again. Okay, so the response is antigen specific. So let's look at the B cell receptor in a little bit more detail. So it exists in two forms. We call it the B cell receptor, or you can call it immunoglobulin if you like. When it's on the B cell receptor, it is a typical signaling protein. The idea is that it can interact with antigen using its antigen binding sites, and that is then transduced into the cell using these accessory proteins that are associated with the B cell receptor that transduce signals into the cell. So the B cell knows when it's binding antigen. There's another form of the receptor, which is the secreted form. You'll notice this one doesn't have any transmembrane domains sticking out the bottom. So this is a secreted form of the B cell receptor, and that's what we call antibody. So the antigen specificity on this B cell receptor and the antibody is exactly the same. It's just that one is a membrane-bound signaling molecule, and the other one is a secreted protein called an antibody. And this is a very tightly controlled process. B cells just don't make antibody without very careful controls, and that's provided by the T cells and a particular subset of T cells called helper T cells. And so those helper T cells are actually required by the B cell in order to make antibody. And it's a very carefully controlled process. So the key to this is actually held by the T cells, not the B cells. So let's look at B cell development. You've probably seen a figure like this before. All the cells in the blood derive from these hematopoietic stem cells. And there's two arms of the lymphoid, which, as you might suggest, guess, is responsible for the development of lymphocytes. So today we're going to look at this pathway, which is the development of the lymphoid cells into a common lymphoid precursor. And that can develop either into a B cell or a T cell. So today we're going to look at this B cell development. There's actually two stages of B cell development we're going to look at. So this first one is in the red box here. We're going to look at this first. And this is the phase of B cell development that happens in the bone marrow, and it's where the antigen receptor is generated by this process of gene rearrangement. Okay? So the hematopoietic stem cell doesn't express any kind of antigen receptor. It has to be generated by a process of somatic cell rearrangement. It's not like a pattern recognition receptor hardwired into the DNA. It's actually something that's generated as the B cell develops in the bone marrow. So we're going to look at this first. Just an overview. There's two chains in the antibody. There's a heavy chain and a light chain. So the heavy chain genes rearrange first, and if that's successful, then the light chain genes rearrange, and then we have what's called a mature B cell. And that then leaves the bone marrow and circulates around the body through the secondary lymphoid tissues. So let's look at that stage first. So this is the kind of a textbook diagram of what's called the germline configuration of these gene segments. This is actually there are two light chains. They're called lambda and kappa, antibody light chains, and there's one heavy chain locus. And so you'll notice that each of these loci contains multiple gene segments, and those are called variable gene segments, diversity gene segments, joining gene segments, and then constant. So whenever you see a V, that's a variable gene segment. And if you look down the list here, you'll see in that lambda chain there's about 30 of them, 30 different variable gene segments. And then a bit further down, we've got these J, which are the joining segments. You'll notice there's no diversity segments in the light chain, or either of the light chains. This is the heavy chain. We've got about 40 heavy chain variable gene segments. There's about 23 diversity segments. There's 6 joining segments, and then downstream we have these what are called constant gene segments. So it looks very confusing, but these have all really arisen by a process of gene duplication through evolution, and each one is slightly different from its neighbor. And that's actually quite important because it means that when these things are rearranged to make a receptor, we get a variety of different antigen specificities. So that's what the gene looks like after it's undergone this rearrangement process. So here we have the heavy chain, and here we have a light chain. So you'll notice we call these domains now. So instead of being gene segments, this is the protein, this is the rearranged gene. And so this encodes what's called the heavy chain variable domain. And you'll notice it's made up of these 3 gene segments. We've got a variable gene segment, a diversity segment, and a joining segment. Okay? So this process of rearrangement essentially takes one variable gene segment, one variable gene segment, one diversity segment, and one joining segment. So essentially random, picks one of each and joins them all together into a contiguous gene, which encodes the variable domain of the B cell receptor. Same thing with the light chain, except there's no diversity segment. There's just a variable and joint gene segment, and they come together to form a rearranged variable light chain gene. And then downstream of the variable, you have what are called constant. So the heavy chain has 4 or 5 sometimes constant domains. So these are called 1, 2, 3, and 4. And then we have one light chain constant domain. So that's how they look like in the rearranged configuration. And then that's how it looks in the protein that's expressed on the cell surface. So we're using the same colors here. So you'll see that there's two heavy chains. This is one heavy chain here, and there's an identical heavy chain over here. Okay, and you'll notice this is the variable domain up here. It makes the antigen binding site, the antigen binding site up here. So this is one variable domain. Sorry, this is one variable domain. The top bit that's composed of the V, D, and J components, and then we've got one constant domain in the heavy chain here, and then we've got the other three form the FC region. Remember, it's called the FC region. That's the region that is embedded in the membrane of the B cell. And then the light chain is next to it. Again, there's an exact copy of that light chain on the other side. So each of these two, those two antigen binding sites are identical. So we have an antigen binding site over here, which is formed by the variable domain of the heavy chain and the variable domain of the light chain. And then you have another antigen binding site over here, which is identical in terms of antigen specificity. So really, this is two homodimers next to each other, and they're held together by disulfide bonds. Okay so far? Yeah. So in the developing B cell, the heavy chain rearranges first, and there's an enzyme called RAG, recombination activating gene. In fact, there's two of them. What's interesting, actually, these are the same enzymes that the T cell uses. T cell generates its receptor, as we'll see on Thursday, in a very similar way, and it actually uses the same enzymes, the RAG1, RAG2 enzymes. So the first thing that happens is this D to J rearrangement. So the green diversity segments and the J segments are rearranged so that you get a DJ, one D and one J joined together first. And then you get a rearrangement again so that the variable gene segment is located next to the DJ. So you get a VDJ. There's a bit of information here I don't think you need to know too much about, but these variable gene segments can be in two different orientations. They can be in forward or reverse. And if you get a joining between two gene segments in the same orientation, the intervening piece of DNA is just looped out and cut. And so these, what are called excision circles, are actually quite useful diagnostic tools. You can use them to see if you've got rearrangement going on inside the lymphocytes. If they're actually oriented in the other direction, you get a strange looping out effect like this. But the take-home message is that the heavy chain rearranges first, the DJ rearranges first, and this particular stage is called the pro-B cell, and then you get the rearrangement with the variable gene segment. That's called a late pro-B cell, and then that heavy chain is then exported to the cell surface. So this is, remember, it's a signaling molecule. So one of the first things that this heavy chain does is it assembles on the surface with what's called a surrogate light chain. This light chain is not a rearranged light chain. It's just a chaperone, if you like, that allows the protein to assemble at the cell surface. And what happens is this surrogate light chain allows that antigen receptor to try and signal through into the B cell. And if the B cell receives signaling through that B cell receptor through these proteins here called Ig beta and Ig alpha, those are the signaling molecules. If it gets a signal, then the B cell goes ahead and rearranges its light chain gene. If it doesn't get a signal, it rearranges the heavy chain again using the other copy on the other side of the chromosome. And if that functions, then it'll do the light chain rearrangement. And if it doesn't signal, then the B cell dies. And in fact, about only half of the B cells make it through this stage of the process. Okay, so they rearrange the gene, they test it to make sure that the heavy chain is working properly, and if it is, it rearranges the light chain. And once you have a functional heavy chain and a functional light chain, basically you have a functional B cell receptor. So at this stage, the B cell is now a mature B cell, and it's ready to go out into the body. It leaves the bone marrow and it circulates around the body. But at this stage, it only has one or other of these what are called immunoglobulin or antibody classes, or isotypes. we'll come back to this. There's lots of different isotypes, but the B-cell receptor is one or other of these two isotypes. It's either IgD or IgM, so immunoglobulin D or immunoglobulin M. Okay, so the B-cell receptor is one of only two isotypes. There's actually 5 altogether. We'll come to them in a bit. But the BCR is only one of two. And like I mentioned, it's expressed on the cell surface with this heterodimer, Ig alpha and Ig beta. These are signaling proteins that are associated with the B-cell receptor that allows it to signal. The B-cell receptor itself doesn't do the signaling. It's those Ig alpha and Ig beta chains that are associated with it. One other thing we need to mention is if you took all of the sequences of the variable domain, the rearranged variable domain, and you align them all up and looked, you'll actually find there are 3 regions where all these differences locate. So those regions are called hypervariable regions. If you line all those sequences up, you'll find 3 regions that are called hypervariable regions, or they're also called CDR for complementarity determining regions. The bits in between are much more conserved in terms of sequence. They're framework regions. And it's probably easiest to understand if we look at this. This is a ribbon diagram of the protein, how it's folded when it's assembled properly. So those 3 loops, as you can see here, there's CDR1 here, CDR2 here, and CDR3. Those 3 loops, when you actually locate them on the folded molecule, are actually right on the edge of the antigen binding site. And in fact, it's those loops that interact with the antigen. Those framework regions I was talking about, they basically form the rest of the domain here, and those are structurally conserved between different antibodies. So the variable bits are actually the bits that need to interact with the antigen. It makes perfect sense. Just one note of interest, though, is the CDR3 loop. This right here. This is actually the product of what's called junctional diversity. And the significance of that will be more apparent when we come to look at the T-cell receptor. But just to sort of put that away and file it away in your memory, CDR3 is the product of junctional diversity. These two, CDR1 and CDR2, are actually inherited in the B gene segment, but the CDR3 is generated during the rearrangement process, which is actually quite important, and we'll come back to that. So why go to all this bother? I mean, we already know that we have this huge repertoire of receptors, and that's how the innate, sorry, keep getting you switched, but that's how the adaptive immune system is able to recognize every pathogen out there. You can make antibodies to any protein, I mean, you can do it to chemicals. You can actually make antibodies to things that don't even exist yet. There's always an antibody in the repertoire that will recognize an antigen. And that's a phenomenal thing to be able to do if you think about it. If you, as you'll see in a second, if we try to do that with pattern recognition receptors, we'd need more receptors that could ever be encoded in the genome. So the reason we undergo this rearrangement is as follows. If we look at all these gene segments, you can see that if we just look at the heavy chain, if we look at just simple sort of multiplying these numbers together, we get over 6,000 different combinations. We pick one variable gene segment, one diversity segment, one joining segment. There's over 6,000 combinations right there. And then if you do the same thing with the light chain and then multiply those together, we get over 10 to the 7 different combinations. And you can get more than that. I haven't gone into the molecular details yet, but some of the, when the antigen receptor is undergoes rearrangement, extra nucleotides can be added to these junctional regions. So between 2 and 20 extra nucleotides can be added to the VD and the DJ junction. So that pushes the number of possible antigen specificities to around 10 to the 9. So we have 10 to the 9 different antigen specificities, which is actually several orders of magnitude bigger than the genome. Okay, so by undergoing this process of gene rearrangement, we can generate all these receptors, which you could never do if they were sort of hardwired into the genome. So the adaptive system is anticipatory. It doesn't know what it's going to be exposed to, unlike the pattern recognition receptors. These receptors can anticipate anything, any pathogen, any antigen. So just to try and illustrate a little bit about what that means, here is an antigen. This just happens to be an antigen on the surface of influenza virus. It's called hemagglutinin, and it's the target for neutralizing antibodies. And it's got this head region, and it's got this stalk region. And the head region is where all the neutralizing antibodies stick. Okay, and there are lots of different monoclonals that have been made in the lab, and they've been mapped to these regions that are recognized. So actually what these refer to are monoclonal antibodies. So those three little footprints on the surface are what we call epitopes. So each of those epitopes will be recognized by a different clone of B cells. So I've kind of drawn that here in a sort of color-coded way. So when you have a response to a complex pathogen like a virus, it's what's called a polyclonal response. So we have lots of clones, and each of these clones is producing a different antigen specificity, and each of those antibodies will bind to a different epitope. In fact, there's probably multiple clones that bind to the same epitope, but for the case of simplicity, we're just showing three mapped epitopes. Okay, so you have a polyclonal response, and it really is there's a lot of redundancy built into the system, so that you have multiple antibodies sticking to different epitopes on the antigen, and that's all brought about by individual clones. The important thing I should mention is that these are very specific antibodies. So the antibody that recognizes this red epitope isn't going to cross-react with these epitopes. These are very specific antibodies. You remember we talked about specificity of the immune response? Hopefully that's fairly straightforward. So we have the B cells. We're going to look at the second stage of B cell development. Most B cells probably never go through this stage. This is what's called the antigen-dependent phase of B cell development. And this is what causes that resting B cell to become activated and then to start producing soluble forms of its B cell receptor. This is what makes it produce antibody. And this is entirely antigen-dependent. So we talked about antigen selecting the clones that recognize it, this clonal selection by antigen. So those B cells that have antibodies that bind that antigen, they're actually, they undergo this maturation process, the second round of differentiation. And this happens in secondary lymphoid tissues. We'll have a look at that in a second. But there are lots of things that go on in those secondary lymphoid tissues, and in particular in this region where the B cells develop into antibody-producing plasma cells. There's a picture of a plasma cell here. These become B cell sort of antibody-making factories. All they do is they just churn out tons of antibody. And all this happens in the lymphoid, secondary lymphoid tissues, in these specialized structures called germinal centers. So this is antigen-driven development of the B cell. Here's a resting B cell. They're roughly to scale. The nucleus is about the same size in both pictures, but you can see in a resting B cell, there's very little cytosol around the nucleus there. This is a plasma cell. You can see it's absolutely chock full of rough endoplasmic reticulum. And what it is basically just churning out tons and tons of antibodies, secreted B cell receptor. So these mature B cells, they circulate, as I said, through the secondary lymphoid tissues, and they're highlighted on this diagram here. So we have lymph nodes, we have the spleen, we have these specialized structures called Peyer's patches. These are all secondary lymphoid tissues, and B cells, they migrate around the body going through these secondary lymphoid tissues. And it's in these tissues where those B cells will encounter antigen. They don't just encounter antigen floating around in the blood, they actually encounter antigen in these secondary lymphoid tissues. They also encounter T cells in there, which is which are required for those B cells to undergo further development. So how does antigen get into the lymph nodes? So when you you step on a rusty nail or something, and you get these bacteria getting into the wound, those antigens will get into what's called the draining lymph node. This is the lymph node that's closest to the site of the injury. And those antigens will either drain in the lymphatics, or if they're over a certain size, a couple of hundred microns, they'll be picked up by macrophages. Well, actually, they're picked up by a specialized cell called a Langerhans cell, but they will migrate with the antigen into the lymphoid tissue And that's where the B cell will encounter the antigen. So the antigen either drains there in the lymph, or it gets carried there by a Langerhans cell, which is a very specialized form of dendritic cell. So this is a bit of information about how those B cells get in. So here's a draining lymph node, and there's the B cell coming in the blood supply. It pops out into the tissues through this little region here called a high endothelial venule. I don't think I wrote that down, did I? Anyway, it's a high endothelial venule. And this is a specialized region of these capillaries, essentially, where lymphocytes will stick, and then they can sort of creep between the cells and into the tissue. And it's in this region here, which is in blue, that B cell, so the B cell's yellow, they'll encounter T cells or helper T cells in this what's called T cell area of the draining lymph node. And if that B cell and T cell talk to each other, you remember the first slide we saw, if they undergo communication, then the B cells will then leave the T cell area and move into what's called a follicle. There are these B cell follicles, and those B cells will start to proliferate. And they will start to expand and proliferate. Remember we talked about this delay period during the immune response. And that's where the B cell undergoes some interesting physiological changes under the direction of the T cells. And those little regions there are called germinal centers. So those B cells are proliferating away. And as we'll see, they undergo three very important changes before they start to produce antibody. There's an electron micrograph, and you can see all these germinal centers inside the follicles. A bit more detail, if you're interested. These B cells are attracted into the secondary lymphoid tissue by cytokines, specifically chemokines. So chemokines are chemotactic cytokines. And so things like CCL21, which is secreted by tissue cells inside the lymph node, those are chemoattractive for B cells. So the B cells, they stick to the inside of these high endothelial venules, and they'll creep between those venule cells and into the tissue. And then there's additional chemokines that attract them into the T cell areas. So the antigens themselves, like I mentioned, those drain into the lymph node in a slightly different way. So the antigens that are shown in these little red blobs here, these drain into the lymph node through the lymphatic system. So the lymphatic system, shown here in pink, it's the kind of interstitial fluid between all the cells, and it's collected by lymphatic ducts, and those are connected to these draining lymph nodes. They're called draining, obviously, because the lymphatic fluid drains through these lymph nodes. And there are all these macrophages called subcapsular macrophages, and those will pick up the antigens, and then actually they'll transfer it to these other cells here inside this follicle. That yellow area there is called the follicle, and those are called follicular dendritic cells. Here's a enlarged picture of a follicular dendritic cell, and their function is to sort of use those long arms to collect the antigen and display it so that the B cells can interact with it. Okay, so these are follicular dendritic cells, and they're bristling with all kinds of receptors. So here's a diagram. I think you can see this down the left-hand corner, but the antigen is shown as this long sort of red wiggly line here, and the epitopes that are recognized by the antibodies are represented by those little red triangles. So you can see we have an antibody here, which happens to be binding to the epitope on the antigen, and it's being picked up by what's called an Fc receptor on the follicular dendritic cell. So remember the Fc region of the antibody? Remember the bit that originally is anchored into the membrane? There are receptors for those regions called Fc receptors or FCRs. And so if you're lucky enough to already have antibodies to this antigen, then you can take up or bind those follicular dendritic cells will bind the antigen quite efficiently. If this is the first time you've been exposed to the antigen, you may not have antibodies, and so the innate system can opsonize the antigen. You've probably had lectures about complement. So complement is part of the innate system, and it can be triggered in various ways. And there are receptors for complement on the surface of these follicular dendritic cells. And so if your antigen is opsonized by components of the complement cascade, then again you can get very efficient binding of your antigen by follicular dendritic cells. Okay, so this is complement mediated, and this is antibody mediated. Either way, these follicular dendritic cells, as you can see in this third panel here, they display the antigen for the B cell to recognize. Now, this looks a bit complicated, but the take-home message here is that activation of B cells takes more than just antigen. Okay, it's a very carefully controlled process. Obviously, the first signal is the antigen itself, and remember we talked about this BCR being a signal transducing protein. So the first signal is through the B cell receptor. Okay, so remember it's either IgM or IgD, and there's often co-receptors associated, that's shown here. This is actually shown here as CD21, which is a complement receptor. So we've got an antigen here, which is being recognized both by the B cell receptor. You can see it's binding to the antigen, that little red circle there. And in this particular case, we have an opsonized antigen. It's had C3b complement component deposited on the surface of it, and that can be recognized by complement receptors on the B cell. So that actually really enhances the uptake of the antigen. Now I'm going to preempt myself, but just in a nutshell, that B cell is essentially I won't call it a phagocyte, but it can ingest what's stuck to the surface. So once that B cell has bound its antigen that it recognizes, it will take that antigen inside itself and it will break it down into peptides. And this is actually something that we're going to look at in a lot more detail on Thursday, but this is called antigen processing. Okay, that antigen is broken down, and then it's a piece of it, these little red, they don't look like it, but these are actually peptides that are derived from the antigen. So that protein is broken down by enzymes, and pieces of that are then displayed on the surface of the B cell. We're not going to go into a lot of detail right now, but it's displayed on the surface of the B cell in the context of what's called an MHC class II molecule. Like I said, we're going to come back to this on Thursdays. And that antigen MHC class II molecule complex is what the T cell receptor sees. Okay, so here's the T cell receptor. So that's the second signal. The first signal is antigen, and the second signal, which is absolutely critical, is the stimulation that it gets from the T cell. And this is called a T follicular helper cell, or TFH. That provides signal number 2 through this co-stimulatory molecule. But that co-stimulation plus the antigen uptake are the 2 signals that the B cell requires in order for it to become activated. Some antigens are what are called T-independent or thymus-independent. Those are antigens that don't require T cell help. So for an example, a lot of these are very long polysaccharide molecules, like lipopolysaccharide. Those can actually cross-link B cell receptors, and you can get signal 1, and signal 2 is provided by toll-like receptors. So if you ever hear about T-independent antigens, for example, if we use a polysaccharide vaccine against a bacterium that has a thick polysaccharide coat on the outside, there are vaccines based on those polysaccharides. Those are T-independent antigens. They don't have any proteins, so you don't get any peptides produced to display to T cells, but they can still induce antibodies through this mechanism. So as I mentioned, the activation of the B cells requires these T follicular helper cells. And we call them helper cells because they provide help to the B cell. And what we mean by help is 2 things. One is the co-stimulation, which comes through this co-stimulatory molecule, this CD40 ligand interaction. As I say, we'll come back to that on Thursday. But then also the helper cell produces these cytokines. You see some cytokines being produced by this helper cell. So those 2 things together, co-stimulation and cytokines together, provide the help that is required by the B cell to differentiate into, well, and here we've got plasma cells that secrete antibodies, but there are other B cells that come of this. The first plasma cells, they produce rather low affinity antibodies, but there are other B cells in the germinal centers, as we'll see in a second, start to produce much higher affinity antibodies. And then there's the memory B cells. So once the B cell gets signal 1 and signal 2, it undergoes differentiation into different kinds of B cells. The majority of them obviously turn into these plasma cells to produce antibodies. How are we doing on time? Okay. Good. So the three important things that happen to the B cell once it gets activated is secretion of the B cell receptor. That's what we call antibody. We also get what's called class switching. You remember we only have IgM and IgD on the surface. Once it's activated, those switch to other types of antibodies like IgG, IgA, IgE. And then this third one is somatic hypermutation, which is what drives the increase in affinity of those cells. So here's a diagram saying the same thing. The first thing is you get secretion of the antibody from a membrane-bound B cell receptor to a secreted form of the antibody. You get class switching to different isotypes. It could be IgG, IgA, or IgE. Each of those have different functions. And then you get this, what's called affinity maturation, process of refining the antibody so that it gradually accumulates mutations to make it a higher affinity antibody. So the secretion is very easily achieved. It's achieved by a process of differential splicing. So when you have the rearranged gene, this is the rearranged gene. It has, you can see the VDJ over here, but the constant segments are upstream of two membrane, transmembrane domains. So when it's a membrane-bound immunoglobulin or B cell receptor, it expresses those two, shown here in yellow, those two transmembrane domains, and those are what anchors in this case, the heavy chain to the B cell. But to secrete that antibody, it actually removes those transmembrane domains by a process of differential mRNA splicing, which I'm sure you've heard about. So this is a way you can take the primary mRNA and splice out those exons that encode the transmembrane domain. What you wind up with is the same heavy chain, but this time it doesn't have the transmembrane domain. And so you'll see it has a carboxyl terminus, but there's no transmembrane domain. That's the first thing. So class switch recombination, or CSR for short, this is the process by which you go from either IgM or IgD to these other isotypes, IgG, IgE, or IgA. Okay, and that's produced by a process that's very similar to the gene rearrangement process that generates the variable domain. So all of these constant domains, which are color-coded here, are downstream of the VDJ. Initially, it will just, in the case of IgM, it will use the mu constant domain, but it can actually rearrange and place that rearranged variable domain alongside either one of the gamma constant gene segments. That will make it gamma is obviously IgG, and then we've got epsilon for IgE and alpha for IgA. So by a process of rearrangement, very similar to the process that generated the variable domain in the first place, you can get that variable domain alongside a different constant domain, and that changes the class or the isotype of the antibody. The important thing to remember is that the antigen specificity remains exactly the same. All we've done is we've just changed the constant domains. Okay, so you can switch from IgM to IgG, for example. It'll have the same exact antigen specificity, but it will now be functionally different because it has a different FC domain. So we talked about IgG and IgM. Just to go back to this diagram, IgM is the one that's produced first, so that that's the one that you find the majority of resting B cells have IgM as their BCR. And of course, once that's secreted, the majority of the antibody that's produced is actually of the IgM class. But that is soon replaced by IgG, and IgG is in fact the dominant isotype that you find in the blood. So IgM appears first, and then IgG comes later as it undergoes class switch recombination. And if you have the secondary exposure, the IgM is the same as it was in the primary response, but the IgG this time is a much bigger response because memory cells are cells that have undergone this class switch recombination. So memory cells usually express IgG or IgA or IgE, one of the class switched isotypes, and not IgM. Okay, so the memory cells produce massive amounts of IgG on subsequent exposure. So the third thing is actually very important. This is the fine-tuning now of those antibodies, and it's a remarkable process called somatic hypermutation. So there's an enzyme called activation-induced cytidine deaminase, or AID for short. That deliberately introduces mutations into the B gene of the antibody. And it's only expressed in B cells in germinal centers. And what that does is it generates a variety of different, say you have one clone of antibodies, but there can be all sorts of different variations of that particular antigen receptor, that antibody. Some will have mutations that make it lower affinity, and some will have mutations that make it higher affinity. And it's the ones that have the higher affinity antibodies that actually survive better. And the reason for that is those are better at collecting antigen and processing it and presenting it to T cells. So the ones that have the higher affinity receptors get more feedback, positive feedback from those helper cells. The lower affinity B cells with the lower affinity antibodies tend to die off. And we can actually see that happening. This is kind of a textbook example of the mutations that occur in the V regions of both the heavy chain and the light chain. And you can see this is after day 7. You get a few mutations, but after a boost, so this is a secondary response, we get a lot more mutations. And then even after a third exposure to the antigen, we're sort of driving out these high affinity antibodies. And you can see that by looking at these sequences, and you can see the mutations accumulate in those CDR loops as complementarity determining regions. And that, over time, increases the affinity of the antibody, which is a remarkable process, really. And that process is called affinity maturation. So once the pathogen has been cleared away, what's left then as most of the plasma cells die off? They only have a short lifespan. What's left of the long-lived plasma cells and those memory B cells? So let's talk a bit about what antibodies do. So antibodies don't do a whole lot, to be honest. I mean, they're essentially an adapter molecule. The only time they do anything of any use is when they neutralize something like a toxin or a virus. And what that means is that they will physically prevent that virus or that toxin from interacting with its receptor on the cell surface. So that's called neutralization. And that can be accomplished by the antibody all by itself. And because it has two antigen binding sites, you can actually get cross-linking of the antigen. You can get large complexes, they're actually called immune complexes. So those immune complexes are usually swept away. They stick to red cells, and the red cells have complement receptors, and they take it to the spleen, and the whole gets broken down. So antibodies can neutralize toxins and viruses, but for the most part, what they do is mediated by the FC region. So these FC regions, they're coded by the constant domains, and those functions are slightly different depending on whether it's IgG or IgA or IgE. In fact, there's a table here. We're not going to go into this in a lot of detail, but these are the 5 main things that antibodies do, and all but neutralization is mediated by this FC domain binding to FC receptors. So there are FC receptors on phagocytes. Here's that opsonization. Okay, so here's a phagocyte, it's a macrophage, it has these little green FC receptors. And if you have a bacterium that's been opsonized by antibody, that really enhances its uptake by a macrophage because it has receptors for the FC region. So opsonization, that's the process by which you cover a pathogen with antibody or complement as well. 2 more things here we don't have a diagram for, but NK cells are kind of like T cells, so they kill virus-infected cells and tumor cells. So NK cells have a way of killing cells if they're opsonized by antibodies. So it's called antibody-dependent cell-mediated cytotoxicity, ADCC. And then there's another one, mast cells have FC receptors. They only have FC receptors for IgE. You may have heard of mast cells in your innate lectures. They secrete histamine, and histamine allows smooth muscle contraction and that kind of thing. It's actually a defense against parasites and worms. If you get infestations in the gut, histamine release allows the gut to expel those worms. And so that's mediated by IgE in mast cells. Okay, if you overproduce IgE, you get allergies. Okay. And then finally, we've got the activation of complement, which again is mediated by the Fc region of antibodies. That's called the classical pathway of activation. Okay, so these are all the different types we talked about: IgM, IgD. In the human, there are 4 subtypes of IgG, all slightly different functions. IgA. IgA is the one that's secreted into extravascular fluids like mucus and tears and milk. And then IgE, which is the one you find in the gut that's immunity to worms. How these differentiate into these different subtypes largely depends on the kind of cytokines that the B cell is exposed to. These are T cell derived cytokines. So we're going to come across this on Thursday, but in the mouse there are a couple of T cell subsets called Th1s and Th2s. And if you're going to try and remember any of this, just remember that Th2s make IgG1 and Th1s produce IgG2. Okay, so those are actually they have slightly different functions, and that's determined by the cytokines that the B cell encounters when it's developing. So just a couple more minutes. Monoclonal antibodies, we talked about these earlier. Remember, serum is polyclonal. In other words, it has a variety of different antigen specificities, but you can make clones of B cells very easily these days from humans, from mice, and so on. And a clone, obviously, is a population of cells derived from a single cell. So if you have a monoclonal population of B cells, they all make the same antibody. And these are very useful, not just as tools in the lab, but they're very useful therapeutic agents. And in fact, a therapeutic monoclonal antibody is now $1 billion industry. And there's a table here from the Janeway book that just shows you some of the kinds of monoclonals that are made. And a lot of them are against pro-inflammatory cytokines, which are designed to bring down inflammation and autoimmune diseases, that kind of thing. So I'm not going to go into any detail. I just want you to be aware of the fact that there are lots of therapeutic monoclonal antibodies out there. And the terminology seems a little baffling at times, but there is actually a method to the madness. So a fully mouse monoclonal ends in OMAB, and then there are these other variants. The problem with using mouse monoclonals is you're going to make antibodies to the mouse antibody, because it's a foreign protein. So the idea is to try and humanize it as much as possible, and it's gone through various stages called, you can make chimeric antibodies. You can remove the framework region of the antibody, replace it with with human antibodies domains, or you can just try and keep the CDR3 loops of the mouse monoclonal, replace the rest of it with human antibody, or you can make a completely human monoclonal. And of course, these are much more desirable because they're perceived as self, and you don't generally get an immune response to them. So I'm just going to finish up with this. We're going to talk about this on Thursday, but no, not on Thursday. We're going to talk about this in November. We're going to look at autoimmune diseases. Immunodeficiencies. So immunodeficiencies are kind of like nature's knockout mouse. There are all kinds of lesions in the B cell developmental pathway. There's a whole variety of B cell immunodeficiencies that we're going to look at. And in fact, the most common immunodeficiencies, these are inherited primary immunodeficiencies, are actually B cell immunodeficiencies. So IgA immunodeficiency, IgM immunodeficiency, and so on and so forth. And we're going to revisit this. Just as a final note, because those individuals are unable to make antibodies properly, a lot of the things that antibodies do, which is to fix complement on the surface of bacteria. These individuals are often very susceptible to certain species of bacteria that are absolutely dependent on antibody-mediated complement fixation. And those are generally the ones that are coated with this thick polysaccharide capsule. Like I said, we'll come back to that. That's my last slide. I just wanted to say that there are other immunodeficiencies that affect T cells and B cells. In fact, there are defects in the RAG enzymes. We talked about those enzymes. Those are the ones that cause the gene rearrangement in the B cells. And in fact, because they're involved in T cell gene rearrangement, you get severe combined immunodeficiency. So individuals that have defects in RAG1 or RAG2 have B cell and T cell immunodeficiencies. That's called SCID. And those kinds of immunodeficiencies are absolutely life-threatening. For B cell immunodeficiencies, you can always do transfusions and give antibodies and antibiotics, that kind of thing. But for SCID patients, they're life-threatening without immediate hematopoietic stem cell transplants. Okay. So I was going to go through the summary slide just quickly. So, so the B cell receptor it's a membrane-bound signaling molecule. It's got two chains, two heavy chains and two light chains. It's produced in immature B cells by a process of antigen receptor gene rearrangement. Each B cell has conferred on it a unique antigen specificity. So these receptors are called clonally distributed. And once they've acquired the B cell receptor, they undergo trafficking throughout the secondary lymphoid tissues, hunting for antigen. And most of them never even encounter antigen. But when they do, they solicit help from these very specialized T cells called T follicular helper cells. And that help is provided in the form of co-stimulation and cytokines. Once they get that help, they then move into the follicle where they start proliferating, produce this thing called a germinal center, and that causes your lymph nodes to swell up. And three things happen. It secretes the antibody, it undergoes class switching to a different isotype, and undergoes somatic hypermutation. So the affinity of that antibody that's secreted gradually increases. And that's it, really. Long-lived plasma cells and memory B cells stay behind and provide that phenomenon called memory, which is unique to the adaptive immune system. Okay, I'm going to leave it there. Hopefully that wasn't too overwhelming. Sure.
Just going back to the dendritic cells, when talking about is it attached to the FC receptor?
So it doesn't just stick non-specifically, actually, like you said, it uses FC receptors or complement receptors.
21.
Oh. So in this diagram, it's actually using both. So these cells, they're bristling with complement receptors. And, it has, they have other receptors like lectins, which will bind polysaccharides, which is a fairly non-specific way. So even if there's no complement fixation or no antibodies, it will still stick to the surface of these cells through the function of, there's a variety of different lectins that can be used. But it's far more efficient, of course, if it's optimized in some way, either by position of complement or.
And then for macrophages, are they attached to the lymph?
Oh, these just roam around. They're in the subcapsular space. They're not actually in the lymph. They're actually in the. They're drawn here, sort of half in and half out. So I guess they probably are able to, foray into the lymph. But they spend most of their time actually in this subcapsular space.